Hello biochemistry students, welcome to your last biochemistry presentation. I would like to congratulate all of you for getting to this point. I'm as excited as you are probably to get this all done. Today I'm going to be talking about regulation of gene expression in eukaryotes. I will probably be comparing them to prokaryotes a little bit along the way, and one of the last things I'll be talking about is RNA interference, which is pretty cool. And here we go. So same DNA, different cell types. How does this happen? The answer is regulation of gene expression, or the process of expressing different genes within each cell, depending on what kind of cell it is. The genes that are expressed actually determine the proteins and RNAs that it contains, and transcription is actually one of the main steps of this regulation of expression. Initiation of transcription. Like I said, transcription is a crucial point for regulation of gene expression, and the ground state is the activity of promoters and machinery without regulatory sequences. This is different in bacteria and eukaryotes in a way that bacteria is considered non-restrictive and eukaryotes are restrictive. So the eukaryotic and prokaryotic ground state differences. In prokaryotic, the RNA polymerase has access to every promoter and can bind or initiate transcription without activators or repressors present. This is considered non-restrictive. While eukaryotic ground states, promoters are inactive when their regulatory proteins are absent, and this is considered restrictive. So eukaryotic versus prokaryotic ground states of transcription. There are four differences that distinguish the regulation of gene expression in eukaryotes from bacteria. The first being the chromatin structure restricting promoter access and therefore transcription to continue. So eukaryotic versus prokaryotic ground states. There are four differences in the regulation of gene expression in eukaryotes versus prokaryotes. The first being the chromatin structure restricting promoter access. So on the right hand side I have a cartoon of a condensed or an active chromatin structure, and I've highlighted an inactive gene, and this is comparative to the relaxed or active gene after modification in order to promote transcription. And the second difference is positive and negative regulatory mechanisms in eukaryotes, which prokaryotes do not have. Positive being the predominant form of regulation. Every eukaryotic gene requires activation to be transcribed. And on the right-hand side, I have something I'll be talking about later, but the initiation complex at the promoter site, and this happens after modification of the chromatin. Third difference is multimeric regulatory proteins. This has to do with size. So eukaryotic cells have more complex multimeric regulatory proteins than bacteria. And on the right-hand side, I have the prokaryotes versus eukaryotic activation. So here we just have like one activator protein, an RNA polymerase, and this is when transcription is on versus the repressor protein turning it off. And in eukaryotes, this is its ground state, which is off because it doesn't have, like I said, this is non-restrictive and this is restrictive. And this is the complex that it takes to initiate transcription. And the fourth difference is the separation of transcription, which happens for eukaryotes in the nucleus like we know, and translation happens in the cytoplasm. On the right hand side I've shown the different cells, and you can see in the bacterium as it's being transcribed, it's also being translated into proteins, whereas in the eukaryotic cell it has to be they're clearly separate, right? Because transcription happens in the nucleus and translation happens in the cytoplasm. and this helps for better regulation in eukaryotic cells. So active versus inactive chromatin structure. We have the heterochromatin, which is indicative of the blue structure on the right-hand side with the inactive gene. This represents 10% of the chromatin at any given time. It's more condensed and transcriptionally inactive versus the euchromatin, the remaining 90%, which is less condensed. So active chromosomal regions are distinguished from heterochromatin in at least 3 ways, and those are the positioning of the nucleosomes, the presence of histone variants, and lastly the covalent modifications of nucleosomes. These are the net effect of so these all constitute chromatin remodeling, and the net effect of the remodeling is to make chromosome segments more accessible to chemical modification, and this is to facilitate the transcription factor activity to regulate gene expression in one or many genes. So there are 5 families of enzyme complexes that actively reposition or displace nucleosomes at the same time hydrolyzing ATP, and this is to facilitate the transcription or expression of genes. 3 of these are important for transcriptional activation alone. So the first enzyme for activation of transcription is SWI or SNF, and this is the switch sucrose non-fermentable enzyme found in all eukaryotes. It has 6 core polypeptides that remodel chromatin. It stimulates transcription factor binding, and I've listed the human analogs of BAF and PBAF. And SWI SNF is actually known as a tumor repressor. They've noticed that it's actually absent in lines of cancer. On the right-hand side, I have a 3D representation of SWI or SNF. And the second enzyme that's important for activation of transcription is NERF. I could not find a picture of NERF, so I've listed the closest thing, which is a Nerf gun. So Nerf actually stands for nucleosome remodeling factor. It is part of the imitation SWI, one of the families that I listed earlier. It is highly conserved among eukaryotes. It remodels chromatin in a way that complements or overlaps the activity of the enzyme I just mentioned. And it interacts with transcription factors to initiate transcription. It's crucial for preparing chromatin region for transcription by catalyzing nucleosome sliding. And it's essential for the expression of homeotic genes, which have to do with anatomical structures. We remember talking about this in genetics, referring to like Cox genes, where legs were being grown on the Drosophilus head when there would be mutations in regions. So this is relevant to that. And the third enzymatic repositioning of nucleosome enzymes is SWRI1. It's the third chromatin remodeling complex that I'll be referring to. It promotes the ATP dependent exchange of histone 2A for the variant H2AZ into nucleosomes. It creates a more transcriptionally competent chromatin state and a boundary for heterochromatin spreading. So here's a list that the book gives us of the enzyme complexes that catalyze chromatin structural changes that initiate transcription, and this may be helpful for some of you. It was mostly helpful for me. So histone modification. So if you don't remember the core histones, H2A, H2B, H3, H4 are modified by methylation of lysine or arginine residues, or phosphorylation of serine or threonine residues, ubiquitination or simulation. I listed the modification type, how it affects transcription and the histone or location that it is modifying it. And it shows you here that the acetylation, methylation, phosphorylation are relevant to small chemical groups, and the following two are larger peptides related. So I'm going to start with methylation. During methylation, the H3 core histone is methylated at its lysine 4 and lysine 36 in nucleosomes near the 5' end of the coding region and throughout the coding region respectively. So the 5' end coding region of the lysine 4 and then throughout the coding region near lysine 36. Lysine 9 of H3 is often methylated in heterochromatin 2. And on the right side, I have the condensed heterochromatin, and it says here that methylation of DNA and histones causes nucleosomes to pack tightly together. Transcription factors cannot bind to these DNA when they're in these positions, and this makes the genes inactive and the genes are not expressed. Here's the little methyl groups in green along the little histone tails. And this is the gene being blocked, DNA inaccessible, gene inactive. HATs and HDACs. Methylation actually facilitates the binding of histone acetyltransferases, which acetylate lysine residues where chromatin is being activated for transcription. There are two types of HAT proteins, depending on whether they are active in the cytosol or in the nucleus. The nucleus has type A, whereas the systolic HATs are type B. Type B acetylates newly synthesized histones before they are imported into the nucleus. And the assembly of histones into chromatin after replication is facilitated by chaperones. The chromatin assembly factor 1, or CAF1, assembles H3 and H4, and the nucleosome assembly protein NAP1 assembles H2A and H2B. And when transcription is no longer required, the nucleosomal acetylation is reduced by histone deacetylase, or HDAC. And this is general gene silencing. This was easy to help you remember, like HATs acetylation lead to the relaxation and active form of the chromatin, whereas the DACs deacetylate the chromatin and you get the heterochromatin or condensed and active form. So positive regulation, I had mentioned earlier that one of the differences between prokaryotic and eukaryotic transcription regulation is positive and negative regulation in eukaryotes. Positive being the predominant only because it's more efficient. Negative can be redundant as far as regulation goes. Eukaryotic RNA polymerases have little affinity for their promoters, and initiation depends on multiple activator proteins, and this keeps from expressing any genes that are not necessary or doing them accidentally. Most eukaryotic promoters are positively regulated. I had mentioned this a little bit earlier in the beginning that eukaryotic cells undergo positive and negative regulation, and that was one of the differences between their cells versus the prokaryotic. So eukaryotic RNA polymerases have little affinity for their promoters. Initiation really depends on the multiple activator proteins being present and in the right positions. Positive regulation is predominant because it is more efficient, and storage of DNA within chromatin renders promoters inaccessible, so genes are silent without other regulation. Specificity for activation is improved when several positive regulator proteins are needed to bind particular DNA sequence to facilitate gene activation. It's a way for ensuring that only genes that are necessary at that time and in that particular state are being expressed, and not just randomly being created when they're not needed, because that would be inefficient for the cell. So on the right hand side, I listed positive regulation. So you have your activator bound to your operator, and this initiates transcription, versus when there's no activator, transcription is not active. Versus negative regulation, so we have the repressor on the operator, and without the repressor there, it's always being transcribed. So the reason why the book mentions that negative regulation is redundant is because for negative regulation, the cell is having to produce that repressor constantly at a specific concentration to keep transcription of a gene from being transcribed, versus positive regulation only has to have it produced at that one particular time in a certain concentration. So it's more efficient for the cell to be positively regulated. Different kind of activators. Polymerase II, as we know, RNA polymerase II synthesizes messenger RNAs. Many, but not all, polymerase II promoters include the TATA box and initiator, not imitator. Sequences with their standard spacing, they vary in number and location of additional sequences required for regulation of transcription. Additional regulatory sequences called enhancers are generally bound to transcription activators. So here we have the transcription activators in purple and the enhancer regions in like navy blue. Other sequences include the upstream activator sequences, UAS. So upstream we have here, and these can be hundreds or thousands of base pairs upstream from the transcription start site. These increase transcription nearby promoters regardless of the DNA orientation. Successful binding of RNA polymerase holoenzyme at promoter requires the transcription activators, architectural regulators, the chromatin modification and remodeling proteins, which I had already mentioned, coactivators, and the basal transcription factors, which I will be getting into all of those in a bit. So transcription activators bind to enhancers or the upstream activator sequences to aid transcription. Requirements for activators vary greatly from one promoter to another. Some are known to facilitate transcription hundreds of promoters, or only a few. Many are sensitive to the binding molecules, providing the capacity to activate or deactivate transcription in response to their cellular environment. So not synthesizing any proteins that aren't necessary at that time to keep everything efficient. Enhancers bound by activators can be distanced from the TATA box. So here I have the activator binding to its site, and then on the DNA, and then I have the activator helping the transcription factors and the RNA polymerase assemble so that we can transcribe the target gene. And on the right hand side, I have the enhancer region like before, bound by the activator to initiate transcription. The second type of DNA binding activator that facilitates assembly of general transcription factors is the architectural regulators. They help activators function at a distance by intervening by the DNA being looped. So looping is promoted by these regulators. They are abundant in chromatin and DNA with limited specificity. The high mobility group, or HMG, high mobility being the electrophoretic mobility in polyacrylamide gels. So it is this domain here wrapping around the DNA and causing it to loop so that the initiator transcription factors can bind and initiate transcription. Coactivator protein complexes. Most transcription requires additional protein complexes, as I mentioned earlier. These act as intermediates between the transcription activators and polymerase II complex. A major eukaryotic coactivator is the mediator. It's 20 to 30 polypeptides long and highly conserved. It binds tightly to the CTD, or the carboxyl terminal domain of the largest subunit on polymerase II. It's required for both basal and regulated transcription. It stimulates phosphorylation of the CTD by TFIIH. The coactivator complexes function at or near promoter's top box. Transcription activation choreography. From the beginning, activators bind to the DNA first, so the activators here bind to the enhancer regions that we had mentioned. The activators then recruit histone modification or nucleosome remodeling complexes, and a coactivator, which is the mediator in this case. The mediator facilitates the binding of the TATA binding protein, or TFIID, and TFIIB, and the other basal transcription factors allow the polymerase II to bind, and the phosphorylation of the CTD region, or the carboxyl terminal domain, leads to the initiation of transcription. The remodeling of chromatin takes place in stages due to these interactions. They are facilitated by interactions between the activators, the hats, like I described earlier, or the enzyme complexes, SWI and SNF. Activators interact with the mediator complex, and the mediator then provides an assembly surface for the first TATA binding protein, or the TFIID, then the TFIIB basal transcription factor, then other pre-initiation complex components, which include the RNA polymerase II. The mediator stabilizes the binding of polymerase II and its transcription factors and the formation of the pre-initiation complex. Most promoters require a precise order of these assembly to initiate transcription, and this process is not always fast. Sometimes it can take up to minutes or even days. The reversible transcriptional activation is more rare. The regulatory proteins that bind to RNA polymerase II, promoters or interact with transcriptional activators as repressors. This inhibits the formation of the pre-initiation complex, which I have on the right-hand side here, that interacts with the activators and chromatin remodeling factors by the mediator complex. When the mediator includes inhibitory units, it also blocks initiation. This is used to ensure a precise order of assembly of the pre-initiation complex, or it deactivates the promoters when transcription isn't required anymore. The minimal pre-initiation complex is not enough to initiate transcription. It doesn't usually form if the promoter is obscured by the chromatin. Positive regulation is imposed by the activators and coactivators. Some activators will adopt different conformations and then act as repressors. A good example of this is some steroid hormone receptors function in the nucleus as transcription activators by stimulating transcription of certain genes when a specific hormone is present. When the hormone is absent, the receptor protein reverts to repressor conformation, preventing the formation of the pre-initiation complex. Transcription activators have domains for DNA binding and one for transcription activation. There are 3 types of structural domains used in activation of transcription activators: Gal4P, SP1, and CTF1 are the ones I'll be talking about. The Gal4P has a zinc-like structure in its DNA binding domain near the amino terminus. It also has 6 cysteine residues that coordinate with 2 zinc ions. It has a separate domain with mostly acidic amino acid residues, and experiments knocking or substituting these let them determine that this is critical to its function, those acidic residues in that domain. The SP1, its DNA binding site is the GC box, which includes the sequence GGG-CGG. DNA binding is near the carboxyl terminus of this and has 3 zinc fingers. 2 other domains for this activator are used for activation, 25% of which are residues of glutamine, which is common for activator proteins. In part P, we see the chimeric protein containing the DNA binding domain of SP1 and the activation domain of CTF1, activating transcription in the GC box region. So many eukaryotic mRNAs are subject to translational repression. Transcription rated in the nucleus must be processed and transported to the cytoplasm before translation. Obviously, this is more in eukaryotes than bacteria, where the two processes are tightly coupled. This imposes for eukaryotes a delay in protein synthesis, but when protein production is needed quickly, the translationally repressed mRNA already present in the cytoplasm can be quickly utilized and translated. The translation of long eukaryotic genes in the million base pair range are regulated this way too. Some genes are regulated at both transcription and translational stages. Translational regulation being like a fine-tuning of cellular protein levels, the binding of translational repressors, RNA binding proteins to 3' untranslated regions or 3' UTR regions of the mRNA. The protein interacts with initiation factors or with ribosome to prevent or slow the rate of translation. So the four types of translational regulation in eukaryotes, the first being translation initiation factors that are subject to phosphorylation by protein kinases. As you've seen before, the phosphorylated forms are the less active, and this causes a depression in translation within the cell. The second translation regulation is that some proteins bind directly to the mRNA and they act as repressors, specifically in the 3' untranslated region. These proteins interact with other initiation factors bound to mRNA or the 40S ribosomal subunit to prevent the initiation of translation. The binding proteins disrupt interaction between the initiation factors, in this case EI, F4E, and E. So here we have the translation repressors binding to the initiation factors and stopping translation. So post-translational gene silencing is mediated by RNA interference, and which is a process that was first established by Craig Mello and Andrew Fire. In eukaryotes, small RNAs or microRNAs or miRNAs mediate gene silencing. They function by interacting with mRNAs in the 3' UTR region, which result in either mRNA degradation or translation inhibition. This forms a regulation control in developmental timing, also used to protect against RNA viruses, especially in plants that lack an immune system. MicroRNAs are temporarily present during development, are referred to small temporal RNAs or sTRNAs. These precursor RNAs are usually 70 nucleotides long, and they have complementary sequences that can form hairpin-like structures. These precursors are cleaved by endonucleases, Drauscher and Dicer, into 20 to 25 nucleotides long, which bind to mRNA to inhibit translation or degrade RNA. These can act on a single mRNA or multiple mRNAs. So over here we have the precursors and the small temporal RNAs that are cleaved by the endonucleases, Dicer, and they go back to silence this mRNA, which can lead to degradation or just translation inhibition. If a duplex RNA molecule is introduced that corresponds to an mRNA, the dicer cleaves the duplex into the small interfering RNAs or siRNAs. These bind to mRNAs and silence it, and this itself is known as RNA interference. RNAs that are produced in the lab have been known to block HIV and poliovirus infections in cultured human cells, and this is kind of a huge field of study right now for inhibiting these pathologies. Woo! We did it! Good job, you guys! If you made it to the end of this video, I congratulate you. Thank you everyone for watching, and I will see you tomorrow. Good job, guys! We did it!